Hi there. Welcome again to our series of short snippets on hematology for medical undergraduates. In this video, we will discuss about the normal physiology of bleeding. And in this video, you will learn about how injured blood cells together with 1 Willebrand factor, platelets and fibrinogen interact following vascular injury to create a primary hemostatic plaque, and how coagulation factors are activated through the intrinsic and extrinsic pathways following tissue factor release to form a fibrin plaque that secures the bleed. The endothelial cells separate the rest of the blood vessels from the flowing blood. Endothelial cells are naturally antithrombogenic, meaning that they prevent activation of platelets and the coagulation cascade from forming a clot within the blood vessels. They are somewhat like a cellophane tape that covers the blood vessels and nothing otherwise sticks on it unless damaged. Intact endothelial cells inhibit platelet adhesion and coagulation activation by expressing vasodilatory nitric oxides and anticoagulants such as prostacyclin, tissue factor pathway inhibitor, thrombomodulin, and heparin-like proteoglycans. However, in the event of vascular injury, endothelial cells can become activated and release 1 Willebrand factor and tissue factor that initiate platelet adhesion and coagulation. Subendothelial structures would include cells such as fibroblasts and smooth muscle cells, as well as matrix substances like collagen, also promote platelet activation and coagulation activation. Of particular importance in the initiation of coagulation is 1 Willebrand factor. 1 Willebrand factor is a large multimeric glycoprotein that is produced constitutively by endothelial cells and platelets. The large 1 Willebrand factor produced in the vibropalate bodies of the endothelial cells is cleaved by a protease known as ADAMTS13 to smaller multimers that circulate in blood. 1 Willebrand factor contains binding domains for collagen, platelets, and factor VIII. The second important player in primary hemostasis are platelets. Platelets are non-nucleated cells that are produced by cytoplasmic fragmentation of megakaryocytes. They are biconvex, discoid-shaped cells resting in circulation, but they, however, assume a spheroidal shape with projections on activation. Platelets also contain various procoagulant factors that are released on activation. Platelets also express receptors on their surface. They can bind to 1 Willebrand factor and fibrinogen. The GP1B9 receptor binds to 1 Willebrand factor, while the GP2B3A receptor has fibrinogen as its ligand. Fibrinogen is the next player in primary hemostasis. It is a circulating peptide that can cross-link platelets by binding to the GP2B receptors on platelets. Fibrinogen can be cleaved to fibrin monomers that can polymerize to form fibrin strands, which is the culminative step as far as the coagulation pathway is concerned. When there is vessel injury, the endothelium is denuded with exposure of the subendothelial surface. 1 Willebrand factor is activated on exposure to the thrombogenic subendothelial structures as well as under shear stress. They are then able to bind collagen in the subendothelial matrix via its A3 domain. They also change their configuration from the tightly packed form to an elongated structure, which would allow platelets to bind to them as they flap around in the circulation. The flowing platelets bind to the 1 Willebrand factor strands by attaching to its A1 domain with the platelet GP1B9 receptor. On attachment and activation of the platelets undergo shape change to a spheroidal shape, and develop pseudopodia to enhance their aggregation. The membrane surface of the platelet also undergoes a process of flip-flop, where the procoagulant phospholipids, which is mainly composed of phosphatidylserine, which usually is on the cytoplasmic side of the platelet membrane, is now brought to the surface of the platelets. This allows for assembly of various coagulation proteins on the platelet surface. The platelet fibrinogen receptor GP2B3A also goes into an active form. With the platelet fibrinogen receptor GP2B3A assuming an active form, fibrinogen is now able to bind to the platelets. Both ends of the fibrinogen bind to the GP2B3A receptors on adjacent platelets to cross-link them. The resultant meshwork of platelets cross-linked with fibrin and 1 Willebrand factor anchoring the platelets to the subendothelium form the primary hemostatic plug to stop bleeding. Concurrent with the platelet hemostatic plug formation, the coagulation cascade is activated. On endothelial injury or activation, tissue factor is expressed on the membrane surface of the endothelial cells. The initiation phase begins with the binding of factor 7 in circulation to the tissue factor, forming the highly active tissue factor, factor 7A complex. Factor 7 in complex with tissue factor is almost 1,000 times more active than factor 7 alone. Tissue factor, factor 7A complex primarily cleaves inactive factor 10 to the active factor 10A. The active factor 10A forms a complex with prothrombin and a cofactor, factor 5A. This assembly occurs on the phospholipid surface in the presence of calcium, which facilitates the anchoring of the factor 10 to the phospholipid surface. This is referred to as the prothrombinase complex. Within this complex, prothrombin is cleaved to the active form thrombin. Tissue factor, factor 7A can also activate factor 9 to factor 9A. Factor 9A can then form an intrinsic ternase complex with factor 10 and factor 8A to generate more factor 10A that can cleave prothrombin to thrombin. The main result of this activation step is the generation of thrombin that can contribute to further amplification steps. Thrombin is a key molecule in the coagulation cascade. It is important to amplify the cascade, and this is due to its activation of factor 8 to factor 8A, factor 5 to factor 5A, and factor 11 to factor 11A. The cumulative action of all these activation is the amplification of the cascade and the generation of factor 10A and subsequently thrombin, which is termed as the thrombin burst. The formed thrombin cleaves fibrinogen to fibrin monomers that can aggregate to form a fibrin meshwork to seal the area of vessel injury. In addition, thrombin activates factor 13 to factor 13A. Factor 13A cross-links the fibrin strands by forming peptide bonds between glutamine and lysine residues of fibrin, thereby stabilizing the clot. Common coagulation tests are designed to identify problems that may arise in different parts of the pathway. The prothrombin time, or PT for short, and the INR, which is a standardized form of reporting PT, assesses the extrinsic pathway. Isolated prolongation of prothrombin time is usually caused by factor 7 deficiency. The APTT meanwhile tests coagulation factors in the intrinsic pathway. Isolated prolongation of APTT is associated with abnormalities of deficiencies of factor 8, factor 9, factor 11, and factor 12. Factor 12 does not usually participate in physiological control of haemostasis and therefore was not mentioned earlier during the description of the coagulation cascade. Both intrinsic and the extrinsic pathways converge at the common pathway where both prothrombin time and the APTT, which is partial thromboplastin time, can be prolonged. Prothrombin, factor V, and factor X deficiencies affect the common pathway. The thrombin time, meanwhile, is a test to detect fibrinogen, and it is prolonged in cases where the fibrinogen is low or abnormal. So in conclusion, the key learning points from this video would be that 1. Wille Brown factor interacts between the subendothelial collagen and platelets to bring the platelets to the site of vascular injury. 2. Activated platelets mediate hemostasis by release of their content, exposure of their phospholipid surface for coagulation factors to bind, and promoting platelet aggregation by cross-linking with fibrinogen. With regards to the coagulation cascade, tissue factor binding with factor VII initiates coagulation through the extrinsic pathway leading to activation of thrombin, and the coagulation cascade is amplified through coagulation factors of the intrinsic pathway, causing a thrombin burst, and the thrombin burst base causes the conversion of fibrinogen to form a fibrin meshwork that seals the site of injury. Thank you for listening.